Hey guys, Tanya and I are here at the Creation Research Society Conference 2025. Look at this. What do you think? Are these those look like smart people, Tanya? What do you think?
I'm not sure if I fit in, but yes, we are here.
We're gonna ask them to dumb it down for us just a little bit. Oh, they got a whole bunch more people in here. Let's walk in there.
What are having?
Here's what I'm thinking, Tanya. We need to do some inspections. I'm a little worried. This is a bunch of scientists. We're gonna have to check out their eyebrows and find out if their eyebrows are scientist eyebrows. But this should be really, really cool. I'm excited about it. So every year they do this conference, and it is. Oh, look at this! Look at this!
Oh, there's a lot of people here.
Wow. So every year they do this conference, and I mean, you're hanging out with just like. Here's David Reeves right here. This is. Oh, I'm not allowed to. Some more scientists right here from ICR. Like, oh my goodness, you're here among. There's Nate Loper up there, and there's like. Guys, okay, they're about to get started, but I can't wait to bring you some information. You're gonna have to just like translate everything these scientists say. Okay, well, maybe we can give you the Tanya translation. After, would that be cool?
I don't have the eyebrow. I don't fit in with the society here.
Well, this ought to be really cool. We're excited to tell you what's going on at Creation Research. What's going on in the cutting edge of creation research? Let's find out.
Welcome to the Creation Today Show, where we bring together interviews with experts and solid Bible teaching. Your host, Eric Hovind, affirms the ultimate authority of God's Word, the truth of creation, and why it matters to you.
Favorite talk so far?
Nate Loper's talk was incredible.
The talk by Dr. Galuza.
My favorite talk was with Dr. Nayeon Lee.
The one I was just in, the Babel one, was very fascinating to me.
Like the geology field trip was a lot of fun.
I like what Joe DeWeese is doing with the dinosaurs and the DNA.
I like Joe DeWeese's talk. That was pretty cool. The dinosaur petroglyph actually being a map.
But I think my favorite talk was called Slaying the Sauropod.
All the scientific findings declare that he's growing. All of them.
Tim Cleary, because he's a friend of mine, but second best, really close, is the Babel dispersion. I thought that was really interesting.
They're all great, but Nate Loper's talk on the petroglyph dragon, natural bridges, I thought was really, really fun.
That's funny. I was actually going to say the same one.
See, I really enjoyed Mr. Bill Hosh's plenary presentation last night. That was a lot of fun.
Favorite talk?
The one thing I learned was that if Eric Hovind doesn't record it, it never actually happened.
Ladies and gentlemen, the new president of the Creation Research Society for a whole year.
Director.
Already. Ladies and gentlemen, the new director of the Creation Research Society, Dr. Joel Brown. Dr. Brown.
Hey.
What a privilege to be here at you guys' event this week. It's been absolutely incredible. If I can just brag on it for a second. You made it where I can't decide which talk to go to because there's so many good ones. Interviewing people, asking what their favorite talk was across the board. I mean, it was across. There was one that stood out, but that was before the end of the day. So I thought, wow, you guys have done a great job.
And that's a credit to all of those individual presenters. So I'll let you be behind the curtain for a moment. We had a 52% acceptance rate on our abstracts.
Wow.
So that means you saw 36 presentations that were here. There were approximately 70 that were submitted. And we are able to select people who are really putting their heart into their research. And that's a good sign in creation science when we have so many people involved and we're able to push ourselves to higher quality research. And that's what you see happening.
Wow. Well, and every presentation I attended was phenomenal. You sat back and you went, this is explaining a lot. They're asking questions that, you guys have no clue the questions that these creation scientists are actually asking. You're like, you wouldn't have even thought of the questions that they're trying to address. Where were you at? Because you did your presentation on specifically the baraminology, the kinds.
Okay, yeah.
So what kind of questions are we trying to answer there?
Well, so our lab focuses on baraminology, molecular baraminology, and molecular paleontology. So we had a presentation in each of those categories. In baraminology, it's a simple question that's very hard to answer, and that is, how do you break up all living things into divinely created kinds? And so we focus on cacti because we live in the desert. And there are a lot of cacti around.
Plenty to study.
Yeah, and who doesn't love a good cactus? So, yeah, we're trying to figure out which of the cacti are actually related because they're all part of one created kind. And probably most of them are part of that one created kind. But you'll have to attend the conference to
see I got to
details.
see the research. It's incredible. Okay, the Creation Research Society in general, I'm a member now. I'm a lifetime member of the Creation Research Society. I'm excited about the future of creation science. How many people do we need in the Creation Research Society? All of them? 8 billion? Like, how many people do we need doing this? How many more people could we use? Where are we at, and how many more people could we use?
Hey, God is just looking for willing servants. That's what we're looking for. And this is really a God-directed mission that we're on. So we're not trying to recruit everyone. We're trying to recruit people who, in their heart of hearts, just want to serve God, but serve God through science. So if that's you, then you should be a member. That's as simple as that.
That's really cool. You guys have some exciting projects coming up. Do you have one that you're more excited about, or just you?
Oh, yes.
Okay.
I'm not going to lie.
All right.
Okay, so, and I'm happy to talk about it. So we've been talking about proteins in dinosaur bones and soft tissues in dinosaur bones. The next natural question, especially as a geneticist, is what about DNA? And everybody wants to find DNA in dinosaur bones, but to an evolutionist, there's no possible way it could be there.
Yeah, no way.
And because it's 66 million years old. DNA doesn't last that long. And they're right. It does not last that long.
It would not work.
But the flood would have buried these dinosaurs 4,500 years ago. So under the right conditions, there is DNA out there somewhere, dinosaur DNA out there somewhere. That's what I would predict from a scientific perspective. It's out there somewhere. And we've got a great team of CRS members that are pursuing this. Wow. And so being able to coordinate all the different efforts from these members is what I find most exciting.
So we very well could in the next however many years actually have some sequenced dinosaur DNA.
We very well could. That's what we're working towards. Now, I'm a scientist, so I have to be completely realistic. And realistic means it's not going to be easy because DNA is fragile. So most of the DNA is long gone. But under the right conditions, which would be a very dry condition or a very cold condition, DNA is still out there, I believe.
Whoa. So if they have a dinosaur bone that they found frozen somewhere, this would be, you guys are ready to do some research.
If you've got a frozen dinosaur bone, let me know.
Let's send it to the Creation Research Society. I love it.
Okay, I have a question for you.
Oh, okay. Switch roles here.
Yeah. So can you describe to just a general viewer, what is the difference between a technical conference like this and ordinary creation conference, now that you've experienced it?
All right, so when I go to a layman level conference, I'm getting a lot of this is the ore, and when I go to a conference, I'm getting just some gold nuggets. I'm getting the tips of, and this, and it's more conclusions. Here I'm seeing all the behind the scenes. I'm seeing areas where in Christian love, there are scientists that are disagreeing, going, I think this is what happened at the flood. I think this is what happened at the flood. I think this is how we should categorize this. I think this is, and so I'm seeing behind the scenes, and I'm seeing the loving headbutting of ideas.
Good way to describe it.
Which is what we want in science. I sit back and I go, this is a good thing, and you are shocked by the genius of the people that are presenting. Like this,
I feel like a dummy around here.
No, these are no slouches.
Yeah.
Like this is not your, I got this wacky idea, let me come. This is well researched, solid, deep understanding of their field and then their presentations. And I find myself in awe, going, God, thank you for taking these minds and using them for giving them the willingness to use them for your glory, because it's really incredible.
Yeah.
It's really incredible.
So I like how you said is kind of behind the scenes. That's a really, can I borrow that?
Yes, you may.
That's a really good way to think of the technical creation research versus the apologetics side. This is behind the scenes. Another way I like to put it is, as research scientists, we focus more on questions than answers.
That's true.
So we are looking for the toughest questions that we don't understand yet. And because of that, sometimes you can come away from a conference like this with more questions than answers. Whereas in a lot of other conferences, you are trying to come away with more answers.
I cannot tell you how many times in the Q&A, you guys gave 15 minutes at the end of every session just for Q&A. How many times did I hear the speaker go, great question, we don't have that answer yet. And I'm just going, there's more creation research we gotta do. Like, that's just, let's get it done. It's pretty amazing. How slow is research based on funding? Is this a, because I look at all these things that's okay, we need more there, we need more there, we need more there. Is this just a matter of manpower, or is it a funding that can provide the manpower? How do you relate the funding of creation science? Because you're not getting government grants to go study creation. If you wanted to study lesbian dance, you could have a grant. But unfortunately, you're trying to study how God created the world.
Yeah, we wouldn't approve that abstract.
Yeah, you would not. Yeah, I'm saying the government would give you funding. How is that relationship, funding to research?
Yeah, I mean, there is a direct correlation. Right? Okay. This is a dollars game in some respects. And there is also a need for people, but I actually think the funding is the first need, because I could hire, I know of 4 or 5 people, qualified people, degreed scientists, we could hire tomorrow if we had the funding.
Wow.
And so there's a lot of talent out there, a lot of new talent, a lot of growing talent, and create young Bible-believing Christians that are scientists that we could put to work had we more funding. So that is certainly, it's a numbers game.
Wow.
In that respect.
We're just like literally provide and it will come. Guys, can I encourage you to become a member of the Creation Research Society if you have any scientific inkling at all? I mean, there's several out there that you can be a part of. This one I love because you guys really are joining all of the research together. That really
I'd like
is the
to
goal.
think we're the largest umbrella.
Yeah, it is.
That's the goal.
It is big. And then be part by donating to Creation Research Society. How cool would it be to see this thing take off like it needs to? You say research is your middle name, and it takes a lot to do the research.
Yeah. No, we appreciate having people like you here. That's the purpose of these conferences, is to just make these connections and build a better creation movement.
This was an incredibly run conference, incredible speakers. Incredible job as the director.
Hey,
I appreciate it, and the host.
You say too much.
No.
We have a great team.
This has been amazing. Yeah. You guys have done good. Thank you, Joel.
Awesome.
Guys, Nate Loper's talk has been, based on our survey, Nate, the #1 talk of the Creation Research Society conference, and all you're doing is blowing holes in an idea that we've had for the past several years.
Yeah, it is. It's an unfortunate truth, but we sometimes got to do that.
Got to do that. All right, so Nate, what did you talk about at the conference? And obviously people are loving it. They're appreciating you bringing out, let's look at this and be honest about it, even if that means we have to change something that we said.
So, talked about the Kachina Bridge sauropod petroglyph, or dinosaur petroglyph, oftentimes used in creation circles to represent a dinosaur, thinking that man and dinosaurs live together. So here's a depiction of it. Well, it turns out that's not what it's actually depicting. It seems to be depicting a much larger, regional map. And so, while dispelling the notion of it being a dinosaur, it also gives us something of wisdom and intelligence of ancient man. So it's actually pretty exciting still.
And the fallout of this is we don't have any evidence now of humans and dinosaurs, or what's the fallout?
Absolutely, there's no such thing anymore.
No, we got to erase all of that.
Not at all. What we're looking at is, first of all, we don't necessarily need a picture on a wall to have an understanding that God's word is true. And when it talks about dinosaurs and man, we can still hold that to be true. But what we need to do is we need to better scrutinize these things and be willing to let go of ideas or theories and things if they don't really work out and make sense. And I think this is just one of those cases. It doesn't do anything for destroying the notion of dinosaurs and man. I still hold to that very much myself. But I also hold to us being people of integrity and of looking at things that are true.
How are we doing as a creation community? How is Creation Research Society, how are all the creation researchers, the scientists doing?
Yeah, we are posed on some incredible things, I think. We had some talks earlier about new technologies available in research, and today we live in a golden age of digital research, and so we can get hold of accesses and records and things that we can really study like never before. So in the organization of Creation Research Society, with research being the middle name, we have research at our fingertips like never before, and I think it's exciting.
So it's a good time to be a creation scientist. Yes, indeed. Well, thanks for the work you do, Nate.
Thanks,
Look forward
Eric.
to seeing you. We're going to be in Mount St. Helens next.
Yeah, that'll be a fun time. You got to come out with us.
Guys, you're not going to believe it. Dr. Carl Warner is at the Creation Research Society. Doc, how you doing, buddy?
Good. I wish my wife would be so excited to see me like you are. I was like, my wife's never excited to see me.
When you brought home the checks, she had to be.
Yeah, that's true.
Hey, I'm so glad you're here, buddy, and this is home sweet home for you.
Yes, I'm in St. Louis. I live in St. Louis. It only took me 15 minutes to get here to the annual meeting. Everybody else had to come from all over the country and the world, so it was great to be here. And so many smart people. I've never felt so, I'm serious about this, I've never felt so dumb in my life. I just came from this ice core lecture. I know nothing. And there's 30 people there have personal experience with ice cores. One guy used to be flying the ice cores from Antarctica. One guy worked in the Greenland. One guy knows this about this. And I'm just like, I am over my head. Then the same thing with the Grand Canyon. There's 30 people in the room that know things about the Grand Canyon, every detail, this plateau, this. And I'm like, whoa, I am way outclassed here, There's some smart people here.
Wow, it makes me very excited. I was going to ask you, what do you think about the future of creation science and the research that's being done?
I feel like we are on the edge of the greatest revival. And that is because whether you've been paying attention or not, the theory of evolution and naturalism collapsed in the last year or two. The public isn't quite aware of this, but it has collapsed scientifically, intellectually it's collapsed. And so this is it. We are now beginning, because there's no other explanation for us, for life, other than we were created. Wow. All the other fields of science, they're going to have to morph into some other model, but it doesn't match up with what we see in the world today. You can't form DNA naturally, how could you form life? You can't form the universe naturally, so how could the universe form? The fossil record doesn't match evolution, it matches the flood. So, to be quite honest, I'm psyched. I really am.
Yeah.
We're going to have so much work to do because we have so many new Christians coming in because the theory of naturalism has collapsed.
That's a great way to put it. How's you've done so much research on the missing links and supposed missing links. Is there any new research that needs to be done there, or is it, you can kind of tie that up with a bow? There's no missing links, or are they constantly coming out with new stuff? Like, where are we at on that?
So you could just say that most of the missing links that are in the museums today are either fraud-based, or they're inaccessible fossils, or they've been disproved. So if you take out those categories of fossils, there's very few left in the museum where you could say this is bona fide works as a missing link. But it will never end. Even though evolution has collapsed, every day they're going to come up with a new one. But you could just say, just generally, the fossils that are out there don't work. That'd be like Toomai, Homo habilis, 1470, Homo erectus. The list is very long. These things just don't work. But they will never die. They will always always providing the next one, because that's how they're going to make their money. But no, it's over. The package is done,
Nice.
Yeah.
Have you had a favorite talk here?
Well, can I re-ask the question? Can I ask myself my own question?
You can ask yourself your own question.
What famous scientists have I met here that are my heroes? Good thing you asked me that.
That's a great question, isn't it? Yes.
There's a guy here from ICR that predicted 10 years ago, 2015, he said the chimp to human DNA difference isn't 1%, which we've been taught since 1974. It's not 1%. 10 years ago, he said it was 15%. And that's Jeff Tompkins from ICR. And this year, he got proved correct in Nature. It is 15% difference between apes and humans. Like, that is the smartest guy on the planet. And even I had to think, these guys are overestimating these differences, the creationists. But they were spot on. I also met another hero of mine. Her name is Dr. Chang Tan. Now, she was a professor at University of Missouri, Columbia. She had tenure. She was fired from Columbia. Why was she fired? Because she understood that the difference between prokaryotic and eukaryotic cells, the translation and transcription, should have been very close, but there was this gap between bacteria and yeast, which should be the most simple eukaryotic cell. For this, she finds out that evolution is not possible between a bacteria and the eukaryotes. And you know what? She got fired. I can hold her up in great esteem, number one, for figuring this out. And number two, she's like a martyr, although she's still here, she's alive. She loses her job for making a scientific discovery. So I'm glad you asked that question. What heroes have you met?
It's a good question, wasn't it? I knew you'd like that question. I know you'd have something to say.
Oh, you put up with a lot for me.
What would you say, hey, here's what we need to do. Here's what we got to focus on.
Joel Brown, who's in charge of this organization, oh my gosh, I don't know how old he is. He's like, is he 40 or something? I don't know.
We're right under.
All these young people here are writing programs. They're using ChatGPT. They're like light years ahead of me. In fact, I can't even understand what they're talking about. And it's the younger scientists that are here. I wouldn't do anything different. I mean, I would say, man, these guys are spot on. In fact, everybody out there needs to donate some money to these organizations and to Eric too.
No, Creation Research Society, baby.
Yeah.
Amen.
The Creation Research Society needs money to implement these great programs they're doing. I was very humbled this week. I thought I would be coming here that I was very smart with creation science. I was like, oh my gosh, I know nothing. Because there's all these fields and all these technologies. A bunch of these groups now here today are decoding DNA in samples. You know, they're doing dinosaur samples and trying to get the DNA out of them. They're doing old fossils, old bones, 500-year-old bones, see how long DNA lasts and stuff like that. These organizations are just great. We just need to. We, meaning you, need to give money to these organizations because they could use the money. Yeah.
Guys, it is so cool to see young people coming to the Creation Research Society and not just attending, but presenting. Jake. You just did a presentation here.
I did, Eric.
And this isn't your first time to present at a creation, where they're critiquing what you do. Like, you've done this at other places, and you just came here to do that this week.
Yep. So I presented this a few times this year, and so submitted to CRS and got to do the talk here. It was my first Creation Research Society
Very
meeting,
cool.
So, yeah.
Well, tell us about your talk. What did you get to talk about? What are you researching, and what are you interested in?
All right. So my interest is in the big question in creation biology, which is we believe that an ark landed in Mount Ararat with 2 of every kind of animal and 7 of some of those kinds. And but those are general categories of just kinds. And as those animals get released from the ark, 4,400 years ago, we hit the present, and we have millions of species, which are specific, not broad. They're specific categories. So my question is, the big question in creation biology is, how do we get from 2 of every kind on the ark to the millions of species that we see today? That's, I think, the biggest question in creation biology, in my opinion. And so my research was surveying. It's called a survey. So I'm surveying the secular, or a lot of people don't like to use that word, but contemporary conventional literature, and looking for data and evidence that will support mechanisms that can help things speciate quickly enough to go from the ark to the species diversity that we have today.
And
Yep.
give me the 10,000 foot view.
Yeah,
do we have any,
So
do we have things?
it was shockingly easy to find things in that area. There's a new process out there called contemporary evolution, and this was actually discovered in conservation biology. So there were conservationists who were putting together these 10-year, 30-year conservation plans on how to save species, and they were finding that they had to take into account the fact that species would adapt to conditions so quickly that they had to take that into account in their conservation planning. So it's basically adaptation in such short time spans. Contemporary evolution meaning years to decades. That's what the contemporary means there. And this is something that they're looking at. So again, the word evolution was actually a little stirring at this talk, but the way I put it to some is, creationists generally don't have a problem with evolution if you put the word micro in front of it.
Correct.
And I'm saying we shouldn't have a problem if you put the word contemporary in front of it, because it essentially means the same thing. It's
micro.
It's adaptations that are happening. They're just happening really, really quickly, and it's not enough to get a human from a chimpanzee, which of course
there's still limits.
That's not even our ancestors, but trying to not straw man them. But it's not enough to make those dramatic changes, but it's adaptations that are going to help a kind diversify across the globe. And they're going to need to do a lot of adaptations to fit into those environments. If you look at some of the environmentological studies, we don't think there were that many kinds of marsupials, but marsupials are very, very diverse. So we need a lot of those adaptations to happen to get all the organisms to where they are today, looking like the way they are today, things that look very, very different in the same created kind. How does that happen? That's what I'm looking at. So contemporary evolution is a great example of that. There are examples in cichlid fish and their reproductive isolation, which means the red fish are mating with the red fish and the green fish are mating with the green fish, and they're starting to split off like that. We have birds finding new migration routes. So there's a bird called the European blackcap, and because of human activity, basically one population found a new migration route. One of them migrates southwest, one of them migrates northwest. They end up in completely different places. They eat different foods, they fly different distances, and because of that, they have different beak shapes, they have different wing sizes, and they look different color-wise. Coloration is different. So these are quick adaptations that only happen in a few generations.
Whoa, so
really
we're talking
remarkable.
one bird source that ended up diverging into these two?
Yep.
And would contemporary science call that a new species? Would they look at that?
It depends. Again, we have there's a lot of different definitions of species. So if someone could easily come in and describe one and say, hey, this is a new species, there's such a strong difference between the two populations that they have seen hybridization between the two, and the hybrids don't know where to migrate. They get lost somewhere in the middle, which is a little sad, but very cool to see that they're so divergent, really, that when they interbreed, when they hybridize, which often happens, you have two populations split up, and then there's going to be a little bit of interbreeding. When interbreeding happens, they get lost in between both migrations. Pretty close to 50% right in between both.
That's amazing.
Which is remarkable. And so that amount of difference only happened in a handful of generations, which is remarkable.
Cool. Okay, are you already relating this to any kind of markers or any kind of is it the environment? Is it an epigenetic thing?
Yeah.
Is it
so for that, I had to borrow some from the conventional literature again. There's an idea they have called adaptive radiation. And adaptive radiations get a bunch of different species in a short period of time. They don't expect to see that now because there's certain conditions that lead to that. And there are 4 actually. So really quickly, the 4 conditions are visiting a new landmass or finding a new landmass, utilizing new resources that might just appear somewhere, an extinction event. So their big example is dinosaurs go extinct from a meteorite. When the mammals survive, they speciate really quickly because of all the new resources. And then the appearance of a key innovation. So they had a new kind of mutation or gene adapts, gives them a new innovation, and then they can speciate really quickly. So those are kind of the conditions. And so I borrowed that term, adaptive radiation. I said, what if we have contemporary evolution today? We have adaptive radiation happening throughout the evolutionary timeline, just rarely. But those conditions match the post-flood world so well. I put the adaptive radiation in the post-flood world where everything is a new migration route, everything is a new resource, everything is a new, is just brand new and growing. And I think contemporary evolution is going to be happening a ton in that post-flood world. So here's my thing is, I think we can build a case for this speciation happening really quickly. And I think we can do that without really even knowing what the mechanisms are for that, which is something that evolutionists and creationists have to figure out, is what's genetically actually happening here.
Yeah.
We don't really know, but the thing is, it's not just creationists who are trying to figure that out, because the evolutionists are now seeing what the conservationists are finding, that there's a speciation or these adaptations happening really quickly, and they're trying to come up with those mechanisms too. But what I did with my presentation, I didn't say this is the mechanism, we now know what it is, but I did say we can make a case for rapid post-flood speciation, even if we don't know what those mechanisms are. And that was really the whole talk in a nutshell there for you.
That's cool.
Yeah,
wow.
It is really cool.
Isn't it cool to see young people? How can I ask how old you are on camera?
I'm 24.
24 and single?
I am.
Ladies.
Yes. Okay.
No, I love it, man. You're 24 and you're saying, man, let me see how God can use my mind for His glory.
for
and
sure.
you're actually helping us solve things that the creation community is wondering and creation science is trying to figure out. So I just think that's really cool.
Hopefully it's a step in the right direction.
I love it. Man, Jake, guys, thank you very much, buddy.
Thank you, Eric. Appreciate it.
One of the biggest pieces of research that has been going on in the creation community has to do with dinosaur soft tissue. Ellie has been doing research on this. I'm so blown away. You're at Arizona Christian University doing the research with Dr. Joel Brown on this.
Yeah.
So cool. Ellie, thanks for being here at Creation Research Society. What did you present on today?
So I presented on collagen degradation in various animal bones and how we measured that and just like the data that we got from that, and also preservation experiments that we ran with that.
So you're literally taking different bones and finding out how fast they'll decay over time, and you're literally doing the experiments right there in the lab.
Yes.
This is cool, guys. This is cool. What did you guys find out?
So we found that after a certain period of time, this bone degrades, and we tested it at high temperatures. We then used that to put it into a graph where we could determine what the half-life would be at different temperatures. So we found the half-life at 25 degrees Celsius in various organisms to be varying between 300 to 1,500 years.
For the half-life?
Yeah.
So if the half-life at a decent temperature, what you'd expect in our world today, if the half-life is 300 to what, 5 or 1,500?
300 to, 1,500.
So 1,500. That puts a really short shelf life overall on collagen.
Yeah. So scientists believe that dinosaur bones, the evolutionary scientists believe that dinosaur bones are about 65 million years old. And if we put our collagen bone half-lives into that, then there's no way that that collagen in that bone could last for 65 million years.
Which means?
Which means that dinosaurs are probably not 65 million years old.
And you got the lab test to show this. Okay, what's next? What do you get? Because you guys are asking questions that most people don't even realize need to be asked in creation science. What do you guys need to do next, and where's that at? Because I've heard ideas of iron is what preserved it, and you guys are actually testing. Could that happen?
Yeah, so we tested the iron cross-linking hypothesis, which is what scientists believe to have preserved those bones for those millions of years. But we tested that, and we actually found that it didn't preserve the bone at all in the amount that we put in it. We were doing other testing that did cause some preservation, but we still need to look into that more and determine what the actual conditions would be for that preservation. So would that actually be present, and would it actually cause the preservation that it was? But even then, I don't think that the dinosaur bone is going to last 65 million years.
It's kind of already a slam dunk, and now we're just shoving it in their face with more research.
Yes.
That's a nice way to put what we're doing to the evolutionary community who says dinosaurs lived 65 million years ago. Well, Ellie would say, I've done the test. They didn't. What a cool time to be a creation scientist.
Yes.
And am I allowed to ask your age on camera?
Sure.
How old are you?
I am 21.
She's 21 years old and doing genetic sequencing and DNA and collagen research all in a lab while you're still in school.
Yeah. Yep. I'm an undergraduate.
This is incredible. What would you say to the young person out there who likes the sciences? Would you encourage them to do this kind of work?
I did not think that I would come anywhere near where I'm at in the lab, and I love it. I love every second of it. And like when it comes up to those presentations, I get nervous, but it's totally worth it, and everyone is so encouraging. So if you want to work in a lab, like, please do it. It will blow you away with how amazing God will be in your diligence to Him in that.
That is cool. And I watched her presentation, and she did kill it. It really was awesome. It
Thank
really
you.
was cool. Well, thanks for being here at the Creation Research Society.
Yeah, thanks for having me.
Yeah. All right, we also got Dr. Deweese here, who's been studying you just did a talk on dinosaur soft tissue. Is this like a slam dunk are we there yet? Can we claim that we've got it? Where are we at in the dinosaur soft tissue research?
That's a great question. It's very exciting. Obviously, the work of Mary Schweitzer and all of her efforts over the last 20-some years have indicated that these fossils have proteins and other things in them, even DNA. And so, I came into it just interested in the question of, hey, could we actually get DNA out of fossils? And we had a relatively low-cost sequencing approach, and I said, let's just try it. And so we were able to partner with some other folks who were able to provide us with some fossil samples, and so we're working our way toward it. It's not a done deal. We're figuring a lot of things out. But I think it's going to tell us something about these animals, and that could be really fascinating. So, just from a pure discovery science perspective, like there's, I think, real interest here that, hey, what can we learn about what these animals were like based upon sequencing data, and, can we get that out? So we'll see.
It's been 20 years since Mary Schweitzer kind of released her information.
Mm-hmm. Yeah.
And creationists immediately ran with it. Are we at the point where creation scientists are leading the way in this research because of the implications of what it means, or is there still a lot of secular work being done on it?
Yeah, I would say there's a lot of secular work being done. There are folks that assume there's nothing left, and so why even bother? But those folks, I think their voices are somewhat being overshadowed by the folks who are still pursuing it. And I would say we're not alone. There are other people pursuing this, and I cheer them on. Like, I want to see that, because I think, again, from just a pure discovery science perspective, it'll be great to see, what we can find from this, what we can learn, what's out there. I'm seeing it. It's growing. And again, we're not alone, pursuing these questions.
What are the next steps for creation scientists then? What research are we still trying to do?
Yeah. So at the moment, my particular interest is in just, can we get DNA out of these fossils and can we sequence it? And I think the answer is yes, but there's still ways to go to demonstrate it and validate it. Because we want to be very careful and thoughtful about it. I do think we have some challenges based on the types of fossils we have available. Some fossils I just think aren't going to be preserved well enough for us to get much out of them, and others may be better preserved. And so there's some factors that come into what samples are we using, how are we processing them, and how can we get the most out of them. And then the big questions on the back end is, okay, well, how do we know what we're looking for, and how will we know when we got it? And those are things we're having to work through, and we've got collaborators and others that we're interacting with around these questions to figure out what should it look like, and what should we be looking for in our data. So,
that's cool. Is there, I know that we now have unfossilized dinosaur bones, do you look for fossilized versus non-fossilized, or is it kind of irrelevant, or does it depend on location?
That's a great question. I think ultimately the ones that are more mineralized, let's say, are going to be less able for us to get things out of them. And so the fresher the bone, the better, and even the more recently it's been dug up, the better, I think, is going to be the case. But that remains to be seen, what we'll get out of them. And so we've started working our way back. We have more modern, I say modern, we have bones from, within the last few hundred to few thousand years that we're looking at, and then we're going to these more ancient bones. And so the question is, will we see a difference in them? Will we be able to get something out of all of them? So far we're making progress. But it takes time, and it takes a lot of effort to figure these things out. So
that's awesome.
We're making progress, though.
You've heard a lot of different talks.
Sure.
We have a lot of different subjects being addressed here. Everything from flower petals to dinosaurs to erosion and what happened at different places. Have you heard one so far between just yesterday and we got all day today? But have you heard one that you're going, oh, that was really cool?
There's several really good talks over the last couple of days, and I will say the area I'm most excited about, obviously, is in the realm of kind of biology, genetics, proteins, this realm, and seeing the work, for instance, Joe Brown and his students are doing. He had a great talk on just trying to understand the relationships between different cacti, and can we use that as a way to understand maybe what the original groupings were of these different plants, in terms of their creation. So can we define a created kind, right, as part of the question? And I think we're seeing progress in that. If you think broadly, if you just zoom out, astronomy, geology, some of these fields are way ahead in terms of developing their creation model and their understanding. I think biology, we lag behind, and we're making progress, we're gaining momentum, but it takes a critical mass of researchers and people looking at these things and asking questions from a very specific vantage point. But we're getting to that place where we're seeing more and more of that, and that's encouraging. And so it's giving us more folks to collaborate with, and more folks to bounce our ideas off with and to get feedback from, and that helps the work grow. And so, hearing talks like Joel's just remind me that, hey, there's some really cool work getting up and running, along with other things that have been more long-standing. And so I'm just really encouraged by that.
What makes an event like this that you guys do every year, what makes this unique?
Yeah. I think this is a great moment just to pause and to get folks together and be face to face and have conversations. I think that's one of the good things about conferences, right? Is you get a moment to kind of sit down and be face to face with others and to think together and to ask questions. I do a lot of Zoom-based groups and things like that, and we interact with collaborators via Zoom all the time because it's just not practical always to get together. But there is something to be said for, hey, let's just sit down and talk together and think through these things together, and to get direct feedback really helps us grow what we're doing and develop it further. So,
fantastic.
Yeah, good question.
Very exciting.
Yeah.
Great to be a creation scientist today.
Thanks, Eric. Appreciate it.
We've also got Dr. Frank Sherwin at CRS presenting on...
Well, schistosomiasis and the the incredible physiological changes that occur in this parasite. It's a trematode, which is a flatworm.
We've actually done a show together on parasites, and I got to tell you, that's one of those things that, to me, the evolutionist, the atheist, that's exactly what I would use, is what you talk about, only try to use it against you, and you're turning it right back around. So tell me what's going on with the, what do you call it?
Schistosomiasis.
That one.
But basically, in terms of parasites fit very, very well with the creation science model. We look at an earth that is young. We find that in the beginning, God created everything very good, and that includes no parasites. And so in the beginning, there was no parasites. And so what we find is that after the creation, there was the corruption, Genesis chapter 3 and verse 1, where Satan said to Eve, did God really say that? And then as a result, God cursed the earth, and he cursed the earth with weeds and thorns and thistles. We think maybe mutations started at that time. My field of parasites, I believe that's the origin of parasites, when God cursed the earth. So parasites are really show that there was a net loss of information. And so some parasites are nothing but a bag of reproductive organs with a spiny holdfast, and of course are very, very small. They fit in, for example, the GI tract of a fish. They're called the thorny-headed worms, but they have no other systems. And so when God cursed the earth, he cursed the earth, causing these, what we call free-living animals that were living outside in the environment doing just fine. When he cursed the earth, they lost a lot of their body systems, and then were basically just a bag of, as I say, reproductive organs. And so that's really fits, I think, the creation model very, very well.
How do you research this? What does it look like to come to a Creation Research Society meeting and go, okay, let me tell you guys what I've been doing research on. I've been looking up data on, what's that look like?
Well, so for example, what we find is that the schistosomiasis that I study has a snail, what we call intermediate host, and then the definitive host, that is the final host, is the person, the individual. And so there seems to be something there when it comes to the incredible life cycle. Their life cycle is amazing. It takes a lot of description there, but it has to be a designed life cycle. It just cannot happen by time and chance and natural processes. We find this amazing life cycle is a result of plan and purpose and special creation. Now, as I say, in the beginning, there were no parasites, and so these creatures that later became parasites when God cursed the earth, they had probably maybe a special relationship with animals, maybe even people. It's called mutualism. So they lived with people, maybe on the body or even in the GI tract, but they were beneficial. Maybe they secreted some hormones or some vitamins or something like that. We have to be theoretical because the Bible doesn't say.
Right, because we don't know. We don't have that
we just
data.
don't know. But of course, the evolutionists are theoretical as well. And so turnaround is fair play.
That's right.
And so we look at that as being beneficial prior to the fall. And we believe that the fall happened very, very soon after the creation of Adam and Eve. So Adam and Eve were in the garden. They were real blood and flesh individuals. They were having fellowship with God through Jesus Christ. And then Satan came along and messed things up. Now we feel that it was a recent fall after their creation, because when they were driven from the garden, it was just Adam and Eve driven from the garden, not their family. So they didn't have time to procreate. Also, we look at the nature of Satan. He wants to destroy whatever God has made, so he wouldn't waste any time getting in there and messing things up. So there's a number of lines that I believe are compelling for the creation science model.
That's cool. What kind of questions are creation scientists asking right now that most people, they haven't even thought of the idea of the question, yet alone the question? Can you think of any off the top of your head that it's like, we're wondering this, and most people are like, you're wondering what?
Well, basically, that's my field is parasites, and I study parasites. I discovered a new species in Colorado, called it Acuaria coloradensis. And so it's a nematode, small white worm that lives inside of the stomach lining of a certain species of bird. But again, it goes back to the big question, Eric. What was the origin of parasites? We have various groups of parasites, the helminth, which are the worms. We have the protozoa, which are the single-celled creatures. They all had a function prior to the fall of Adam and Eve, prior to the curse. And we have very interesting indications of that. So when people at the conference say to me, what about the origin of parasites? There is a mutualistic relationship that's possible, but also we find there are parasites today that are what we call free living. They live out in the environment, they don't interfere with people or animals, but under the wrong conditions, people might ingest the ova, that is the eggs, and become infected. One example is called Strongyloides stercoralis, and that is
that one.
Yeah, and that is a nematode, and it can be quite a serious infection, but it is free living as well. And so there's an indication there. Even evolutionists admit the step between a free living worm, just living on its own in the environment, not bothering anybody, and becoming a parasite, even the evolutionists admit, is just a one-step process. So it doesn't take millions of years, it doesn't take hundreds of thousands of years, it just takes one step, one mutation in order to cause a parasitic relationship with an animal or a person.
And you're looking at that one step going, that could be the result of the fall.
That's exactly
the
it.
cursed creation. All of a sudden, boom, now this, wow.
So, and I like to use evolutionary literature to make a point because it fits so very well with the young Earth creation science model.
That's beautiful. Thanks for the work you do, buddy. How you've been involved in the creation science research for years?
That's it. Almost 30 years now.
Wow.
Yeah, and so I was a teacher in Pensacola Christian College. I was teaching a variety of pre-med courses to the students there, everything from medical microbiology to anatomy and physiology, parasitology, just a number of classes, cell biology, and that's the Lord getting me prepared to go out into ICR, into creation science research. And so that was God preparing me, and I really appreciate that time. Plus, 3 out of my 4 children were born in Pensacola.
We like Pensacola, baby. We like Pensacola.
I love Pensacola.
What excites you about the future of creation research? Do you feel like, man, we're just getting started? I mean, 30 years in, I look at the talks we're giving today, these days, and I go, I'm really excited. Is it just because I'm younger? Is it because we are really making some progress here?
I am very excited because even the evolutionary naturalists are seeing that time, chance, and natural processes simply don't answer the big questions about why we have the great variety of life in the world today. It's not because of time and random changes that occur. There has to be plan and purpose and special creation. That is in the area of the biological sciences. That's why I'm excited to see that there are, and this is something I emphasize a lot, there are natural limits to biological change. And that's what I would tell my students in Pensacola. There are natural limits to biological change. You can make all sorts of varieties of roses, but you cannot make a black rose because it's not within the genome, it's not within the genetic makeup. And you can have all sorts of varieties of dogs, lots and lots of varieties, but they're still 100% dogs. Same thing with fruit flies. Fruit flies have always been fruit flies, and of course bacteria like E. coli. There's a guy in Wisconsin, a PhD scientist, who does not like creationists, that's putting it nicely. And since the 1970s, he has been plating out bacteria, E. coli bacteria, right up into the year 2025. And guess what it is? It's still bacteria. And so we're going, thank you for doing all that work. You're making the point for us. And so I saved the best until last, Eric, and that is soft dinosaur tissue. And not only are we finding the soft dinosaur tissue, but we're finding soft tissue of lots and lots of other kinds of creatures as well. So the geologic column, if you get in there, we're finding untidy discoveries, evolutionarily speaking, of soft tissue. And so we're excited about that. We're excited about what happened at Mount St. Helens. That was 30 years ago. With Mount St. Helens, it shows that you can have catastrophic processes happening very, very rapidly, forming geologic features that have the appearance of old age, but we know formed about 30 years ago. Plus the rapid recovery of Mount St. Helens. Used to be, 50 years ago, evolutionists would criticize the creationist, saying, oh yeah, worldwide flood, you'd have the world that was just totally destroyed, and how is it that they were able to recover from that? And that was the same question asked by a lot of evolutionary ecologists after the explosion of Mount St. Helens. It looked like a thermonuclear blast zone, a lunar landscape. And they made the prediction that it would take centuries to recover from that. And of course, we know it just took a couple of decades, and we see lots of animals, lots of plants coming in, repopulating. So these are exciting events that occur in the young Earth creation science community.
So it's a good time to be a creation scientist.
That's it. It's a good time to be a creationist. And so I continue to do the research, and yeah.
I love it. And we need more young people out there doing it.
Yeah, and there are young people taking up places that old people like myself, and so we're excited to see that research and that work being done. And really, when you get into things like molecular genetics, when you get into the very, very tiny details of the individual cell, it shouts creation. Right now, Eric, as we're sitting here, we have submicroscopic protein-based machines that are literally going down and up our DNA spiral helix cells and repairing the lesions and sometimes the mutations that we constantly get by sunlight striking our cells and messing things up in the DNA. That happens all the time, minute by minute. But God, in His infinite wisdom, has designed these protein-based machines that are submicroscopic. You can't even see them, hardly with even an electron microscope, but they're doing an amazing job of replacing damaged nucleotides. A nucleotide is a sugar, phosphate, and a nitrogenous base. That's the functional unit of a DNA molecule. These submicroscopic machines are doing the repair work minute by minute, second by second, all of our lives. And it's just amazing. How do you get those submicroscopic machines' origin? If you had the origin of DNA, the DNA, evolutionarily speaking, would immediately start getting mutated. Where are those protein-based machines going to come from to repair that? Because in order to have that, those protein machines are called editase, like an editor. ASE means an enzyme. So you have these editorial enzymes, as it were, that do the repair work. But if you have the origin of DNA by time and chance, it's a classic egg-chicken paradox for the evolutionist.
And if that's fixing the DNA, that's preventing evolution, because evolution depends on the DNA getting messed up and evolving into something different.
That's a good point. That's a good point. Man. So it's extremely theoretical.
That's cool.
And so we look at it as saying, in the beginning, God. And we make no apology for that, because the atheist says, in the beginning, nothing. So you have these two worldviews: in the beginning, God. We don't make any apology. In the beginning, nothing. And I say it's more scientific to say we came from a creator, a loving, just, all-powerful creator. The evolutionist says, I will not bend the knee. Everything came from nothing, which is a neat trick. And so I always like to gently challenge the evolutionist to say, show me an empirical research that could show something coming from nothing. I'll wait.
Just sit back and hold tight.
Yeah.
Actually, you don't have to hold your breath on that one at all, do you?
That's it. And so let me just say one other thing, and that is people don't understand that the Big Bang does not explain the origin of the universe. The Big Bang only explains the expansion of the universe. So I always like to ask the atheist what happened 5 minutes before the so-called Big Bang. Don't tell me about the Big Bang. And it has a lot of problems with it. We won't get into that. But it only explains, supposedly, the expansion of the universe, not the origin of the universe. And so they're in a quagmire about that as well.
Wow. I love it. Good time to be a creation scientist. Thanks for leading the way.
Okay, thanks, Eric.
Appreciate it.
All right.
We are sitting down with Dr. Anderson, who is with the creation. Are you a doctor?
No, I'm not
I
a
thought
doctor.
you and your husband were both doctors.
Well, actually, I have my PhD, putting husband through.
That's really good.
My father-in-law gave me a certificate when Kevin got his PhD that I got my.
You got your PhD.
I got my
PhD.
That's
beautiful.
Yeah.
Okay, so Dr. Anderson, you guys were with the Creation Research Society for how many years?
20 years.
20 years.
Yeah, I started. Technically, we started a little bit off because we had kids still in school. So he started, and then I came with the kids later. But I officially started in 2004.
Well, I got to tell you, I have been familiar with for several years, but not like involved in or reading or. To see you guys and what you guys are doing, I am very excited about the future of creation research. It's really, really exciting. Tell us a little bit about what it looks like and what you guys are trying to accomplish.
Well, that's a big question. Just to get the message out there to people that you can be a scientist, you can follow what science says and be a Christian. The Bible and science do not disagree. And the funny thing is, you see a lot of times in the headlines, science says. No, scientists say XYZ. And it's like saying, your hammer says, well, no, a hammer is a tool. Yeah. Science is a tool. And then depending on your worldview and how you are looking at the science determines what it says. And so you can have two people look at the same data, one being a creationist or a Christian, and somebody who's not, and get totally different results afterwards. So
you guys have been involved in a lot of different research projects, and your husband specifically worked on the iDino project.
Correct.
What excites you right now about all the stuff you've seen, and when you see these abstracts and where we're going, what excites you about the future of creation science?
Just there's a lot going on, and there's a lot of people doing a lot of really wonderful, good research. And that's kind of the important thing, is that we do good, very, just really on the mark.
Thorough, accurate, make sure we're getting this right.
Yeah, and not just, well, I think maybe whatever. No, we have the data, we have the information, we have what we know is what we have.
Sometimes it feels to me like the creation scientists are still just getting started. It seems like the world of science was born in creation science, hijacked, and it feels like we're just kind of starting to wrestle it back away.
So good to see here at our conference so many young people.
Yes.
It has been wonderful. We've always been laughing that the problem is everybody not aging off, but literally, we're usually the higher in our ages, number-wise. And so it has been so exciting to see so many young people. We had, 2 13-year-old students here today, and that is just really neat to see them and have them be here and be involved, and seem to be enjoying it too. So, but yeah, it's exciting, and we just hope that more young people will understand the importance of getting involved and getting that information and understanding the information and then participating in what we're doing. So
I love it. Well, thank you for the work you do.
Oh, well, thank you.
What a privilege.
Thank you.
Awesome. Guys, it's a great time
to
be a creation scientist.
Yeah.
No doubt about it.
Yeah.
Awesome.
All right. Thank you. Thank you so much.
Don, welcome to the Creation Research Society, bud. What are you presenting on today?
Thank you. So I presented on a possible trigger for accelerated radioactive decay.
This is a huge problem in creation research right now. So give us the background to it, and then tell me how you're trying to solve, what your hypothesis or testing or research is on that.
Yeah, absolutely. I'm obviously a big fan of the RATE team, like most of us are, I think, and so I was excited to see their research come out showing that there was lots of evidence that all this accelerated radioactive decay took place during the flood. The problem was, what do you do with all the heat generated by all that decay?
And the atheists love pointing that out, the skeptics in evolution. Well, hang on, you guys didn't solve it because
of the heat problem. And so almost a decade ago, actually, it really bugged me, and I think I found out at least a plausible possible solution, and that is, if before the flood, the Earth, all the way to the core and mantle and everything, was cool, like room temperature cool, then I did a calculation that shows the amount of heat needed to raise the temperature to the current temperatures is very, very similar to the amount of heat that would be released in the 4.5 billion years worth of decay. So more or less, that's the solution to the heat problem.
So it's possible that the original creation Earth did not have a hot molten core.
Yeah, and there's other reasons to think so too. Like a lot of theologians and creation scientists have thought that there weren't any earthquakes or volcanoes before the flood.
Correct.
And so if a solid, cool Earth would naturally be free of earthquakes and volcanoes.
Does the solid, cool Earth affect the magnetic field?
It certainly could, because at that point the core of the Earth, the iron core, could have been a permanent magnet, and you could have had a much, much stronger magnetic field that wasn't decaying at all because it was just a permanent magnet. So that would do a better job protecting the pre-flood Earth from all those cosmic rays that are currently striking us because our magnetic field is weakening.
And you wrote a paper on this?
Wrote a paper on this in 2012. It's not well known, of course, in the Journal of Creation. That's okay.
That's going to change because both of my friends are going to go read it right now. Both of you guys, go check this out. So 2012 creation, so you can go to Creation Research Journal?
Well, you could. I just went there the other day and they only have back to 2015 on their site, so I don't know how to find it anymore.
Well, we're going to be fixing that.
Okay.
We're going to be working on that, so we'll have that fixed.
Okay, great. So then at the last International Conference on Creationism, I had an opportunity to kind of extend this view. I looked at the moon and Mars and some other planets and moons, and they have giant thermal expansion cracks, actually. And so like on Mars, it's called Valle Marineris. It's 10 times the size of the Grand Canyon. It's like almost a third or a quarter of the whole way across Mars. And so what you can do is you can take these thermal expansion cracks and you can say, well, if it was because of thermal expansion, if the inside of Mars heated up and cause it to grow a tiny bit, well, how much growth would be required in order to make a crack that size? How much bigger did Mars get based on the size of the crack? And based on how much it grew, you can calculate how much heat had to be generated inside Mars to cause that thermal expansion, and you can compare that to the amount of energy produced by the accelerated radioactive decay. And again, there's a very close match between how much heat internally Mars had experienced and the 4.5 billion years. Same thing on the moon. NASA had a gravity mapping mission called GRAIL, and they found like half a dozen or more Grand Canyon-sized just cracks filled with lava. You can't see them on the surface because they're filled with lava, but they found these cracks, and they're not like a round circle like it could have been from an impact. They're just all jagged and angular like a polygon. And so NASA's even saying this doesn't look like an impact to me. And so again, thermal expansion cracks is probably the best solution. Again, you can measure the expansion of the moon based on these cracks, and again, there's a match between the amount of heat inside the moon and 4.5 billion years of decay.
That's fantastic. Wow. Tell me what the creation, what does this do for the creation community?
Absolutely. Well, it's certainly great to get to know other creation scientists and present my ideas. I've had a lot of great feedback from the members of the CRS, actually. So, there were a few weaknesses in my original presentation, kind of hypothesis that I've kind of refined my hypothesis a little bit based on some constructive criticism, and that's been very helpful. And it's a way to just get to know other creation scientists and be encouraged and hopefully encourage them as well.
Amen to that. Thanks for doing what you do, Don.
Hey, thank you.
Yes, sir.
Nice to meet you.
Wow. What an incredible experience. To see behind the scenes of creation research and to see what's actually happening, it's really pretty mind-blowing. Did you have any favorite things? It was, we heard everything from dinosaur DNA to Lucy to rock strata to what impressed you, Miss Translator, on what happened in the world of science?
Man, I think obviously some of the topics were a little bit too hard to understand for me, just because I don't necessarily have like a super scientific mind, which I wish I did. It's such an amazing stuff to learn and to know, and especially from creation perspective, I think that's amazing, just because you're putting God in everything. Well, because God is in everything, so you're just drawing those treasures from every topic, and you're seeing how it goes back to the Lord and how He created, and that's amazing.
And that is how science started. Science was born in the womb of people going, I want to see God's hand in nature. I want to see what God did. So I feel like we're watching creation scientists get back to that, and that's really encouraging for me to see us do research and do science in the womb of giving God the glory for His creation. So I just think it's incredible.
Absolutely. And I think if you have any interest in science and learning, getting to the nitty-gritty things of how it's made, so to speak, just get a hold of this organization and see how they can plug you in, or where they can plug you in, and who you can get in touch with to learn so much of how it's made and how it's, I don't know, it's just everything is just so
questions.
amazing.
The questions that we don't have answered yet are big.
Yeah, and I think once you put so many minds together, it couldn't be a huge, amazing explosion for creation.
I think some of the best things are yet to come. That's what I think. I think they're going to be incredible. Now, I'd encourage you to get involved as well. Check out Creation Research Society. Go to their website, see what they're doing. And Tanya, I don't know if you know this. Do you happen to know where next year's Creation Research Society meeting is going to be taking place? Do you happen to have any clue, or did I
don't know. I heard a little birdie, and I don't know if it's true.
What'd the little birdie say?
Is it true that it could be in Pensacola, Florida?
It's coming to Pensacola, Florida, guys. That's our hometown. We don't have to travel this time. We'd love for you to come visit us in Pensacola, hang out with us at the Creation Research Society. Get details online. It'll be 2026 Creation Research Society meeting 2026 in Pensacola, Florida. Pensacola Christian College is going to be hosting it. I couldn't be more excited about that, to watch Christian colleges get involved and get, stay on the front lines of creation research. We're watching colleges do that, and I love it.
Pensacola is just amazing city to be in anyway, just like the white beaches, the Gulf of America.
We've got a pretty sweet gig
and
there, let me tell you.
Pensacola Christian College is an amazing facility as well.
Yeah,
Come
you're going to
join
love
us.
it. All right, we'll see you guys next year at the Creation Research Society. God bless.